So today we are going to discuss polar amino acids. So amino acids are basically building blocks of proteins, or you can say proteins are actually made up of different types of the amino acids. So basically we categorize the amino acid into two, non-polar amino acids and the polar amino acids. So if you see here, we categorize the amino acid into the non-polar and the polar amino acids. So non-polar amino acid, we have discussed non-polar amino acid in the previous video, but now we are going to discuss polar. So we further categorize the polar amino acid into two, charged polar amino acid and the uncharged polar amino acids. Those amino acids having charge will be categorized in the charged polar amino acid, and those amino acids having no charge will be categorized in the uncharged polar amino acids. Okay, so what actually the group that actually make the amino acid polar? What things that actually make amino acid polar? So two most important groups that actually make the amino acid polar. First group is the hydroxyl group, OH group, and second group that actually make the amino acid polar, that will be the carbonyl group. So here are basically the two most important group, and these group will be present to polar amino acid having no charge. Okay, so what actually these groups do to the amino acids? So these groups actually helpful in the formations of the hydrogen bond. So hydrogen bond is very much important. So hydrogen atom can make bonds with the three most important atoms. That will be the nitrogen, oxygen and the fluorine. Any one of atom, either nitrogen, oxygen or fluorine, can form hydrogen bond with the hydrogen. Hydrogen must be present in the hydrogen bonding. It can be between the nitrogen and hydrogen, it can be between the oxygen and hydrogen, it can be between the fluorine and the hydrogen. So actually if you see oxygen is present in the carbonyl group, then it's mean that that can easily bond the hydrogen bond with the hydrogen atom. Okay, similarly here is the presence of oxygen, and oxygen again easily bond the hydrogen bond with the hydrogen. So it's mean that these OH group, hydroxyl group and the carbonyl group can make the hydrogen bond with the other compounds. So that's why these are basically the polar amino acids having no charge. So we will discuss the six most important polar amino acids. So first one is the serine. Okay, so general groups that are actually present in the amino acid, first one will be the carboxylic group. Okay, second group will be the amino group NH2. Third will be the hydrogen. These three groups will be common in all amino acids. So what actually the difference between the amino acid? So difference will be decided by the presence of the R group. Okay, so in this serine you will see the presence of the one carbon, and at that site you will see the presence of the OH group. Now this OH group will make the serine polar amino acids, but in that case you will never see any charge on that group. Okay, there is no charge present at that group, so that's why we categorize it into polar amino acid having no charge. Okay, second one is the threonine. Okay, so in the threonine you will see the presence of the two carbon chain, and at the first side you will see the presence of the OH group. Now OH group will make the threonine as polar amino acids. Okay, third amino acid, that is the most important, tyrosine. So in the tyrosine you will see presence of one carbon group. Okay, you will see the benzene ring, and at the last you will see the presence of the OH group. Again, these three amino acids, serine, threonine, and the tyrosine, having OH group. Now, OH group will make all these amino acids polar. Okay. Next is the cysteine. That's the most important. You will see the presence of the CH2 group, and at the last you will see the presence of the sulfhydryl group. Okay, or you can say thiol group, or you can say sulfhydryl group, SH. Okay, now H from the sulfur can easily be removed in the alkaline medium. So removal of the H positive easily in the alkaline medium will make this cysteine as polar, and that polar amino acid will be categorized in the uncharged, because you will never see the any charge present at that site. Okay, that's the most important structure of the cysteine. Okay, next is the asparagine. So in the asparagine you will see the presence of the group, that is the CH2. Next you will see the presence of the carbonyl group. Oh, that's most important. Carbonyl group C double bond O. Now this carbonyl group will make the asparagine polar. And last you will see the presence of the amide group. Okay. Next is the glutamine. So in the glutamine you will see the presence of the CH2 group, and last you will see the presence of the carbonyl group. Okay. And at the carbonyl group you will see the presence of the NH2 amide group as well. So, two asparagine and the glutamine having carbonyl group, and the serine, threonine, and the tyrosine having OH group. So that's why all these amino acids are polar in nature. Okay. So important thing which you have to remember, these group can easily make hydrogen bond or can attach other group easily due to their polarity. So if I give an example of this one, threonine. So threonine having OH group. Okay. Now this OH group can easily bind the phosphate group. Okay. So here is the phosphate group. Structure of the phosphate will be this one. Here is the phosphoric acid. Phosphoric acid will be H3PO4. If we remove the hydrogen at that site, okay, that's the most important thing. If we remove the hydrogen, then they will carry the OH negative charge. So now this will become the phosphate group. Okay, now this phosphate group can easily bind at that site. Here is the presence of the phosphate group. Okay, it's mean that OH group, at the OH group you can bind the phosphate group, and that will happen in the presence of the enzyme, and that is the kinase. So kinase enzyme actually helpful in the addition of the phosphate group, or you can say phosphorylations of the amino acid with the help of the kinase. Opposite enzyme will be the phosphatase. Phosphatase enzyme actually remove the phosphate group. So here are basically the two enzymes that works oppositely. Kinase add the phosphate group, and the phosphatase will remove the phosphate group. Okay. And last most important thing, difference between the cysteine and the cysteine. Okay. So if you see here are basically the two molecules of the cysteine. Cysteine molecule 1 and the cysteine molecule 2. Here are basically the two cysteine molecule, and you will see the presence of the thiol group. Okay, here are basically this thiol group of the cysteine 1. Here is the thiol group of the cysteine 2. Now between the thiol group you will see the formation of disulfide bridge, or you can say disulfide linkage. So here is basically the sulfur, and here is the sulfur. Now between the sulfur atoms you will see the formations of the disulfide bridge, or you can say bond. Now, when the two cysteine molecule joined by the disulfide bond, then it will become the cysteine. Cysteine. Here is the Cysteine, and here is this Cystine. So here is basically the different Teine and Tine, cysteine. So this is all about the polar amino acid. If you still have any question regarding these amino acids, you may ask in the comment section. Thank you so much.